my YouTube channel, and anyone can watch it. To go back and review. And I will try to get the slides in as well, although that's not great. Okay. So everyone should have signed up for a pathogen. I think the first presentation is coming next Tuesday. I forget who that is, but okay, thank you. I said that I would give a presentation first to give kind of a sample of what you need to do. So I made a slide. Since I'm normally up here, you don't have to make a slide, but you can make 1 or 2 slides if you want. That's not going to affect your grade. So I just picked chickenpox because I noticed no one had picked that, and it's something that we are all familiar with. So chickenpox is a virus. So the scientific name is the varicella zoster virus. It's highly contagious, as most of us probably know. Now there is a vaccine for it, but there didn't used to be. I think the vaccine came out maybe 20 years ago. If you didn't have chickenpox, you probably still know that it causes a rash all over the body, which has 2 types of symptoms. These little pink bumps, which are itchy, and then there are these vesicles, which are fluid-filled and they burst, and then those are contagious. And then, of course, you have the scabs from these burst vesicles, and then whatever you scratched off, which is bad, and they always say that it's a scar as well. So this is a painful and highly contagious condition. It lasts a few days, but one of the issues with this and a lot of other viruses is that it's actually contagious. You're shedding the virus particles before you show any signs. So that means that even if you stay home after you start noticing the chickenpox, you might have already spread it, and that's also one of the reasons why it's so important to have a vaccine for a lot of these diseases, because you don't know if you're spreading it or not. So this is, as I said, a virus. This is an image or a diagram of the virus. This is an actual micrograph of the virus, a scanning electron microscope. So this virus does have an envelope, which means that it's similar to a plasma membrane. It has phospholipids, and then it also has proteins in the envelope, and these proteins importantly have, sugar molecules on them that form different structures, and these are used to bind to the host cells and then get inside of the host cell. So the inside of the envelope, there is a capsid or a nucleocapsid, and so this refers to what we've talked about in the past in terms of viruses. So I've drawn this on the board before, this little kind of spaceship-looking thing that's made up of proteins. So this is all proteins. And then on the outside of the proteins, you have a lipid layer. And so, importantly, the viruses that have a lipid layer and are called envelope viruses are susceptible to drying out. And so that's why it's really effective to do things like wash your hands. And so they don't live for really long periods of time or anything, because they need to stay moist at least a little bit. So we have the lipid, then the proteins, and then inside of the protein capsid, we have the DNA. So some viruses have RNA, others have DNA. This one is double-stranded DNA, very similar to pretty much any organism that you've studied before. The genome is sequenced. It's a relatively small genome, especially compared to any kind of eukaryote or prokaryote. So it's about 70 genes. So that's many, many fewer genes than humans have. And it's closely related in terms of gene sequence for most of its genes to the herpes virus, the human herpes virus. So as I said, the glycoproteins, which are in the envelope, bind to host receptors. And as I think most people have heard about by now, the chickenpox virus can invade these certain cells, and does invade these certain cells, these neural cells, and it kind of stays in these neural cells. And this is what actually causes shingles, which is when the virus becomes active again in these neural cells, and that could happen throughout your lifetime. So they have that commercial that said, if you had chickenpox, then you already have the shingles virus inside you, which is true because this virus will remain inactive, so it's not going to be producing new virus particles most of the time, but the DNA is still there. It still has all the genes, so it can reactivate and use the host machinery to cause very similar symptoms to chickenpox, but they're usually more localized. So any questions? Yep.
If you get chickenpox, is that the same with the vaccine?
So if you had the chickenpox vaccine, I think that you will not get shingles either, because you are not going to have that DNA. You're going to have some antigen which recognizes probably one of these glycoproteins or one of the other proteins, maybe in the nucleocapsid. So you don't have the genome of the virus to reactivate.
If you had a chickenpox, but you had no vaccine.
I'm not sure. I think it depends on which protein is actually made the vaccine, or which protein the vaccine is made from. Not sure. I'm not sure if you're allowed to even get the vaccine if you have had chickenpox, because I'm not sure if it then might possibly damage the neural cells. I'm just thinking out loud here. I really don't know. I'd have to look more into that.
Shingles vaccine.
Yes, I think so. So it's probably some activator protein that the shingles vaccine is targeting. Any other questions? Okay, so that was a good little discussion of our first pathogen. Okay, so this week and next week we have kind of a lot to talk about, and they're complex subjects, and that's one of the reasons why I'm recording this class, because this goes into biochemistry, and this is really the foundations for all of the molecular stuff that we're going to talk about for the rest of the semester. And the book kind of just goes through all this stuff, and it doesn't give it too much context, so I'm going to try and give things a little bit more context and relate them to microbes or to humans in different cases. But some things we're not going to get to cover. Certain things you should know. So I sent out an email yesterday saying, definitely look over the vocabulary. We're going to use that vocabulary even if I don't define everything in class. It should be stuff that you've seen before. I still recommend brushing up on it. It doesn't hurt. And it'll help jog your memory. So the chapter starts out with this image of the Miller-Urey experiment. So this experiment was supposed to simulate some kind of atmosphere on an early Earth type planet, and then they sent essentially a lightning bolt into here in the form of an electric spark, and they waited and observed to see what would occur and what kind of molecules they ended up with in the reaction vessel. So can anybody tell me the relevance or the significance of this experiment? What did they find and why was it important? Yeah? Right. So this experiment was trying to prove that we could make organic molecules from non-organic molecules, and that the precursors to biological molecules existed on the early Earth. Now they got some things wrong because it was found that the early Earth's atmosphere was not really as similar to these conditions as they previously thought. And a lot of textbooks write this up as, oh, they found amino acids, but they didn't find all 20 amino acids. They found a few amino acids. And what's the other relevant thing about this? Does anybody remember what was non-lifelike about the amino acids? Well, there are no proteins. They are just singular amino acids, like scattered around. So what does the text talk about later in the chapter? The two different forms of amino acids? Does anybody recall this? The L and the D? So what do we predict was created in this experiment? So in this experiment, both of the forms of amino acids were created. So in organisms, we only create one form of the amino acids to use in proteins. In this experiment, it made both types of amino acids. So that could be true, that could be what actually occurred in this early Earth scenario. There are both types of amino acids made randomly, and then organisms just adapted one of them and consistently used one form. Or again, there could have been something else that's missing from this experiment. And so this is just one experimental theory on how life and organic molecules formed on the early Earth. There are a lot of other theories. And what else is missing from here? So, okay, amino acids are important, but what else do we need for life? We have amino acids, which we know will be used for proteins. What else would you say makes an organism? Cell or some kind of membrane? Some kind of lipid enclosure? And then what else? Some kind of nucleic acid, yeah. So while this experiment was very exciting, it did have flaws, and it only hypothesized and answered to part of the puzzle. We still don't know where the other components came from. There are theories for those. We don't have a theory of how it all came together as one. So that's still a real black box in the area of astrobiology and early Earth chemistry. And so, of course, since we're talking about cells and what makes a cell, I'm just going to write on the board, maybe over here at the top, our central dogma, because we're going to be referring back to this at certain points during the class. So our central dogma is that we have a genome, which is made up of DNA, from which we make messenger RNA, and then we make proteins from amino acids. This is our central dogma of molecular biology. Sometimes it's called the central dogma of modern biology. And this gives us a framework for which to think about how cells function. And it shows us our connection between these different types of molecules. So again, we could structure our thinking. Why do we need nucleic acids? How would we have consistently put together amino acids for any function if we didn't have nucleic acids, according to this paradigm? And so we're also going to be talking about macromolecules as well as some smaller molecules in the cell today. We've talked about some of these before, but we're going to kind of categorize them a little bit more, hopefully. So this is our periodic table, and we have certain elements that are much more common in cells than others. And can anyone give me the acronym for the most common elements? Yes, Caitlin. SNOPs, right? CH. It's not schnapps like the brand. It is spelled very differently, but this gives us our acronym for the most common elements in any cell. And I liked that our textbook actually quantified it and said, hey, these make up 99% of the dry weight of any cell. I think that's very telling. So it doesn't mean that you have these atoms individually. They're certainly parts of molecules, but the molecules that are common in the cell are made up of lots of carbon, lots of hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And so I always say, if you don't know what atom is in some biomolecule, a good guess would be one of these guys. Then in addition, on this periodic table, there are also some other things highlighted. But before I tell you exactly which are which, I'm going to ask you, what are some important ions that you've learned about in the past? What are some ions that have been used for proteins and maybe signaling? Potassium. I heard a lot of whispering too. Sodium. Magnesium. Yep, talk about chlorine. Magnesium. And chlorine.
Salty banana is forever stuck in my head now.
Yeah. Salty bananas. Because we have lots of potassium in bananas and salt, as well as chloride. So these are some really common ions that we find in the cell, which you may recall the sodium potassium pump. I'm sure that we all know table salt, sodium chloride, magnesium. All these things also notice, these are things that you find in like vitamins or this makes a complete breakfast cereal or whatever. These are things that we need to consume in our diets. Oh, I wanted to, darn it, this didn't pop up. So the give me 1 second. So I think that I accidentally deleted it, but the So the ions are highlighted in purple, and then the blue element, or the blue atoms, are the trace elements. And so these are things that we need in very low quantities. And when we talk about making media, which we will in the future, you'll also notice that a lot of these things you need to put in the media for bacteria because they also need some of these ions and trace elements in very low quantities in order to grow. Now, on the other side of the coin, some of these in high doses are toxic to both humans and bacteria. So especially different types of metals can be toxic to bacteria, and obviously to us humans as well. And some metals have also been claimed to be antimicrobials, but in small doses, these things are important. So this is just a reminder of some of, again, the structure of some of the Schnapps elements and where their electrons are held. So I like to just give this as a reminder because we tend to only talk about the outer valence electrons, but we should remember the other ones are there. And this also gives us a nice guide to how many elements each one of these is going to bind to. And of course, carbon can bind to, and usually does bind to, 4 other atoms. So that's normally how we see it in any organic molecule, in any molecule that is in any cell. If carbon's not bound to 4 other atoms, or if it doesn't have some kind of double bond so that its entire valence shell is full, then that's usually pretty dangerous, and that can really harm the cell. And hydrogen is another really common element. So many of our organic molecules, the dry mass of them would be primarily composed of just carbon and hydrogen. So the textbook refers to small molecules and common small molecules. What do we mean by a small molecule? Because kind of by definition, molecules are small, right? But when we're talking about small molecules, it's normally in organic things like carbon dioxide, ammonia, and oxygen, or the oxygen, molecular oxygen, where we just have a few atoms bound to one another. Conversely, those macromolecules that I keep referring to are larger molecules that form very large structures that have hundreds or thousands of atoms when put together as a polymer. So all these things are macro structures, amino acids and proteins, membranes, and nucleic acids. And if you think about, imagine that they're much larger than these small molecules here. And so remember, even though normally we think of just organic molecules, and especially these types of molecules in the cell, there are inorganic small molecules which are really, really important. So what are some common characteristics of organic molecules in the cell? I kind of gave some of them away, but again, if you were to guess what atoms are composing or the main composition of some organic molecule, what would you say? Carbon, right. Carbon, and then goes along with hydrogen. And a lot of times we have multiple carbons in a single organic molecule and multiple hydrogens per every carbon. And that's most likely what everyone saw when you took intro bio, thinking about the different metabolic pathways, so things like glycolysis, starting out with glucose. So you have 6 carbon molecules, then you have hydrogens on each of those, and then in addition we also have oxygen, which is again one of these most common elements. So we kind of already gave away the categories of organic molecules in the cell, but let's go through and talk about some of the examples of these, and also we didn't give one important category, which we're going to be talking about a lot today, and which the book talks about. Can anybody tell me what that category is? So what is glucose an example of? Carbohydrate. Carbohydrates or sugars. So these terms are usually used interchangeably. And what is a carbohydrate?
Sugar used for food.
Right, so many times these are molecules that can be broken down to generate fuel in the form of usually ATP. So as we said, our most familiar carbohydrate is glucose. But what are some other carbohydrates that we're familiar with? Glucose. What else do we see on the nutrition panel? Fructose. Yep. What else? Sucrose. Like just replace the first letter. Lactose. So these might be most of the ones that we're familiar with, at least the naturally occurring sugars. Although we're probably familiar with a few of the artificially made sugars, which we'll talk about in a couple minutes. Okay. So carbohydrates, all of them, all simple carbohydrates have structures similar to glucose. But glucose has 2 forms, and that is a linear form, which is shown here, and then a ring form. So we may have seen both of these and not made the connection. The linear form can just kind of circularize, and you can see that the carbons, we have one carbon that's not part of the ring, and then we have oxygen, and then we have the carbons going around, which are each number in the ring. I don't have my laser pointer out. But the other thing that I wanted to mention is that you can have different versions of the ring. So this is, what type of bond is this between the 2 carbons here? It's a covalent bond, and it's a single bond. So what are the chemists always telling us about single bonds versus double bonds in terms of 3D structure?
Single bond is linear, 180 degrees.
So a double bond,
We're talking about just the central bond, it's still linear, it's still many degrees.
well, but how do these carbons move with respect to one another? They can't. So this molecule is flat, it's planar. These molecules up here, the carbons can rotate at every single bond. So you could have each of these OHs and Hs kind of revolving if the axis is going straight down. And so you have them flip-flopping on one side or the other because these single bonds don't hold those into place in terms of turning around.
If that would be like the tetrahedral form, and then it would actually be a chain like that as you go across. You
Yes,
looked at it from the side.
yes. And so that's a good point, just to make sure we're on the same page. I've shown two ways of drawing this up here, but then another way of drawing this would be to say, okay, we have C double bond O, and then we could do C here, and then just this jagged line would be OH. So each of the points where we make a zigzag represents a carbon and infers that whatever hydrogens should be there to fill the carbon's valence shell are there. And this also has an OH. So this would be the same as that, but then the ring structure is separate. So either of these two, the one that I just drew or this up here, these can rotate and they can continue to rotate freely, but then the ring form cannot rotate freely. So if you've taken the top carbon and connected it to the second to the top carbon, connected it to the bottom carbon here, and you're creating a ring structure, and you can no longer flip these back and forth. And so that's why there's a difference between the alpha glucose and the beta glucose. The differences are pretty small, but you can see with the alpha glucose, the hydroxyl groups are below in alpha, and in the beta, they are opposite. So they could be below or above. You can flip this whole thing over, but the difference is that they're on the same side here, and then they're on opposite sides here in the beta glucose. And so once you go onto that ring structure, then that's permanent for the ring until you take it apart. So if we take in organic chemistry in here, no. Well, we'll talk about this a lot if you take organic chemistry. We're not going to go into everything, but this is the reason why, or it reflects how organisms can only use one form of these sugars because they have different structures, and structure is related to function. So what are some of the ways in which cells use carbohydrates? We already said producing energy. What else do we use carbohydrates for in cells?
Receptors on cell wall.
Yep, so we said that they're used to identify and to recognize the different cells. What else? They can be parts of messenger molecules. So what else? What is the cell wall made of? How do we draw the cell wall? So in plants, cellulose. So if we're going to draw a cell wall, what's one of the ways we draw the cell wall? What shape do we draw? Okay, here, guys. It's like all this noise in the hallway. What shape? Triangle, stars, circles. No. Yeah, so we have these shapes and they're repeated, right? And these are actually carbohydrates. So in addition to storage for energy, we also have stored carbohydrates that are used structurally. So that's really important for bacteria and plants, because cell walls are crucial to the survival of these cells. So a little bit about the naming of carbohydrates. When you have just a single carbohydrate, whether it is in the linear form or in the ring form, then they're called monosaccharides, because mono means one. And these are very simple sugars. They include things that we're familiar with, like glucose, ribose, so this is also this might be familiar to us from our nucleic acids. And then there are also other things that we might have heard in chemistry, like glyceraldehyde, dideoxyacetone. So these are just different forms of sugars. And we see, but they all have the same kind of basic structure as glucose. They all have some carbon with a double bond to an oxygen, and then we have a string of other carbons with OH groups on them. Okay, so that's the theme here. And they have different numbers of carbons, and we have the carboxyl group at different places, but we have all those components in all of these sugars. Again, these are all monosaccharides. I'll come back to this in a minute. Oh. Well, ignore the background here, but these are examples of disaccharides. And so we're also familiar with many of these, which are maltose, sucrose, and lactose. And so these are two sugar molecules connected to one another with a covalent bond. And they can have different numbers of carbon molecules and different bonds, but these are all disaccharides, again, because they have those two sugar molecules. Disaccharides are always in the ring form. There's no such thing as a linear disaccharide. So you never have that linear form with two sugars. They always have to be in the ring form. So different organisms make different disaccharides, and different organisms can break down different disaccharides. And again, it depends on the shape of the enzyme and what it can bind to. And I wanted to point out that the bonds here are represented by different shapes. They are both what is called glycosidic bonds. That is, just means bonds between two sugars. So this is glyco. I'm not going to ask you to know the different types of glycosidic bonds, but you can know that there are different types, and this just means that it's a bond between two sugar molecules. And as we've kind of already alluded to, here we're seeing disaccharides, but we can also have trisaccharides and strings of saccharides, which we'll look at in a moment. And again, the two different types of glycosidic bonds cannot usually both be broken by the same enzymes. And so to talk about the enzymes a little bit, so what enzyme breaks down lactose? Lactase. Is that what you're going to say as well? Yep. And so what enzyme breaks down sucrose? Sucrase, right. And maltose is maltase, and etc. So a lot of these individual disaccharides have their own enzyme which breaks them down. And when we talk about breaking down sugars, this is why I inserted this slide about the artificial sugars. So if these are artificial sugars that weren't normally in our environment, would we have enzymes to break them down? No, there's no reason for us to. So this is one of the reasons, or this is kind of the theme of artificial sugars, that they are similar enough to naturally existing sugars that they stimulate our taste buds. So they could bind to those enzymes, coincidentally, but the enzymes in our stomachs can't break them down, so they kind of just pass through and they don't count as palatants. And surprisingly, these artificial sugars can actually activate more taste receptors than some natural sugars, and that's why they just taste so sweet. And you use so little of them that it would be incomparable. Like if you were making a recipe, does anybody cook with artificial sugars actually? Yeah. And how much do you add in a normal recipe?
Depending on the sweetener that you're using, like some sweeteners, like I think stevia drops are like super sweet compared to regular sugar, so you only add a couple drops for like tablespoons worth of sugar.
Right.
Whereas others, like a teaspoon to a tablespoon, it just depends on how sweet it is.
Even a teaspoon to a tablespoon, who else bakes in here?
It's like 8 teaspoons to a tablespoon.
Okay, yeah. So normally you add like a half a cup or a cup of sugar. That's a lot. And so this is actually some of these sugars you can break down, but again, it's kind of not even significant because of the amount that you're using and that are added. So this is pretty neat. It's unfortunate that they have other effects and bad connotations. Otherwise, this would kind of be the perfect solution, right? So the formation of a glycosidic bond is shown here. And you can see that we have the two ring structures, and the glycosidic bond is made when we connect the two sugars at the hydroxyls. So the hydroxyl, one sugar is going to be lost, essentially releasing water, and in addition to the hydrogen of one of the other sugars or the other sugar. So we see the water here, and then we see the glycosidic bond here. So this is one version, and then another version of the glycosidic bond would just kind of be the opposite, so that the oxygen would be up here and the hydrogens would be down here. So this is a condensation reaction. So if we connect many of these different sugars, give me 1 second. I apologize, I don't know why this crazy thing happened where it's showing body so that you can't see anything. There. So if we have a lot of saccharides combined into a very long string, then our sugars are monomers. And what do we make when we connect many monomers? A polymer, right? So these are saccharide or carbohydrate polymers, and there are different types. So glycogen is one of the ways in which we store sugars in humans. And so if you're thinking about going on some of these new fad diets, then what you're aiming to do is to actually access the glycogen and use that glycogen, your stored sugars, as opposed to using your glucose that you've recently consumed in your diet. Then another form of carbohydrate polymer are starches, which are not made by humans, but which we can break down. And then the third one here is fiber or cellulose, and we can neither break down fiber nor create fiber. So that's why fiber is great to have in your diet, because we don't have the enzymes which could break down these molecules. So it creates a lot of volume, you feel full, but it's not really ending up to be calories. I also wanted to point out, so these look really neat. They have all these branches on the glycogen and the starches. So in starches in particular, there are different branching patterns. So branching off of different carbons in the ring, and those require different enzymes. And so there are certain enzymes that are going to cut apart the linear part of the storage molecule, and then there are other enzymes that will clip off anything that is in like a Y intersection. So again, very specific relationship between the substrate and the enzyme. Of course, essentially, everything that we're talking about here is made up of glucose, because all of these storage polymers are made up of the saccharide glucose. Even though we know that there are other simple sugars, none of those are incorporated into this, because why would we store those? We would have to convert them into glucose before using them. Storing glucose as a polymer itself is the most, I think, efficient way to store your energy molecules. So that once you break these glucose molecules out of these structures, then they could be used for energy. Not necessarily in the case of cellulose in plants, because that's part of the structure of the cell, but for the glycogen and the starch. And then this is just a close-up showing in glycogen the different enzymes which are going to break different bonds. So there's enzymes called amylases. So this is a little bit different than the setup we were talking about before, because this is glycogen and it's not glycogenase. I don't know. It's amylase, which breaks down this large polymer. And so amylase comes in two different forms, the alpha and the beta. And we have them working on the different parts of the glycogen molecule. And after we break down the larger pieces off of the molecule, we still need to then, in addition, break apart the single components of the polymer. So we still need to break off the glucose molecules one at a time, and then you need to break down that glucose molecule. And that's all before you even get any energy out of this whole thing. Okay, and again, don't know why this is being all weird.
I think it said as the background to your slides.
Yeah, but I modified the main thing and it shouldn't have done that. Oh my gosh. So we're just going to put a shape in there so that we could read what I've written. So we talked about energy, we talked about structural uses for carbohydrates. And in addition, we've talked about identifying cells and recognizing cells using carbohydrates. In addition, there's a relationship between carbohydrates and pathogenicity. So that is related again to this identifying cells. So there are these carbohydrates called, it's a class of carbohydrates called amino sugars, and then a type of amino sugar is the nanomolecule. So this is called an amino sugar because it has an amino group here. And I don't need you to know the structure of this, just need you to know the background and the significance of this. So this molecule is made by certain pathogens, and it's displayed at the very end of one of these glycolipids. So you might have a long chain of saccharides. So we got some pathogen molecule here. So it might have, we might have a chain of molecules, and we could have some branching on this as well, similar to our storage molecules. Then at the end of this, it puts up this nanomolecule, because this is the one that it wants the host cells to interact with. And the host cells can't tell that this is a bacterial sugar. If the host cells saw these other sugars, then it would be able to identify this as a pathogen. But for some reason, the host cells don't recognize, either can't bind to the nana, or is a wrong enzyme or protein binds to the nanoparticle. And so this nanoparticle also helps the pathogens adhere to the host tissues. So many times pathogens are particularly dangerous, not because they're swimming all throughout your body, but because they are forming a colony in one particular part of your body, in one tissue or one surface. Just like in particularly bad infections, they can even look like the things that we grow on plates in the lab. So looking at a colony that you can even see visibly with the eye in certain instances of very, very bad infections. And so this nanoparticle helps the pathogens to start settling down and adhering, holding on to that tissue. Humans, on the other hand, have an amino sugar, not the nanoparticle, but something similar to that, which we naturally incorporate into mucus, and that helps to stop bacterial growth. So it really depends on the exact structure of the saccharide, and that correlates to its function. So we have two similar amino sugars, one which is used by bacteria to fool the host, and then another one which is used to prevent bacteria from actually settling down and forming a community. So that's how it combats bacterial growth. Any questions so far? So I'm not going to talk about sugar alcohols, really. But I will talk about enantiomers and chirality. So I think that we already brought up enantiomers. And I'm not sure if we actually use the word isomer, but isomers are molecules which have the same components. So that they have the same atoms and the same number of atoms, but you could have different structures. And so the different sugars that we were seeing before were isomers. So these three sugars are isomers: glucose, galactose, and fructose. they all have the same number of carbon molecules, the same number of oxygens, the same number of hydrogens, but they're arranged differently. So if you were just given the chemical formula, you might not immediately be able to discern them from one another. So again, those are isomers. Then there's a subset of isomers which are called enantiomers. And they specifically are even more similar than most isomers. Same number of carbons, same number of oxygens and hydrogens, but in addition, they have the same structure except that they are mirror images. And so this is what will result in the D and the L form of different ring structures for sugars. And so that's different from what we talked about before. It's not the alpha and the beta sugars, it's D and L. So these are both alpha sugars, alpha glucose. And you don't need to memorize all of this, but you should know that in our metabolism, we use only the D form of glucose. And the D and the L come from the original mode of classification of these molecules, and you don't need to know the background of that or how the light is refracted or whatnot. You just need to know they're two different structures, they're two different enantiomers. So I know that everybody always loves chirality when I teach it. It's like the most favorite thing in intro bio. And personally, it was my favorite thing when I took chemistry. So I know we need to review it. So we were talking about chirality on this previous page. So whenever you have an enantiomer, two mirror image molecules, then one of those atoms is going to be chiral. So in this case, which carbon is chiral? And we start numbering. This is number 1. Five. Number 5 is the chiral one is this one that stems, or which the OH groups, which are different, stem off from. And so many times, if you want to identify a chiral carbon, you just look for the carbon that's bound to 4 different groups. And this is why we usually talk about chirality in terms of amino acids, because this is a carbon here at the center of this cross. It is called a central carbon. And in an amino acid, any amino acid except for glycine is going to be chiral or have a chiral carbon because it's connected to 4 different things. It's connected to the carboxyl, the hydrogen, the amino group, and the functional group, or the R group, whatever you want to call it. So these 2 molecules right here are mirror images of one another, and in reality, they're not superimposable. And what do I mean by that? Well, I have a nice video, but it means basically they're like a glove or a shoe. They are not interchangeable. You can't put a shoe on the wrong foot. And if you have gloves that have the things on the palms, then you can't put them on the wrong hands. It just doesn't work. So this video is pretty nice because it just shows you how they're not superimposable. So again, you see the mirror images. The central carbon is connected to 4 different things. Even if you turn it, these 2 things match up, these 2 things match up, but then the front group is an amino group in one of them, and then the other one it was the hydrogen. And you could go back and look at this. Like this is the link here, it's on YouTube. I know that some students just like to really watch this in order to cement the concept. So we're talking about chiral carbons and organic molecules. There are also non-organic molecules, like down here, this bromofluoromethane. This is another chiral molecule, which we don't care about so much right now, but just for your background knowledge, there are non-organic molecules that have chirality. Now, going back to our amino acids. And so now you know the difference between the D and the L form of amino acids. And so you can start to get an idea of why in that Miller-Urey experiment, it was significant that both types of amino acids were produced. So we don't know if at the beginning there were both types of amino acids, and again, organisms only used one, or if only one type of amino acid was produced. It seems like, so there are arguments as to whether DNA came first or amino acids and proteins came first. It would seem like if DNA came first, then the amino acids would then be coded for in the DNA, and then you would end up getting only one form of amino acid. But that's not what we saw in this experiment, so we don't know. The book was a little bit cryptic on this point, and to me, it wasn't clear about the fact that there are no L amino acids in proteins ever. Some organisms can use these forms of amino acids for signaling. So they're just used as signal molecules, but they're never incorporated into proteins because, again, they're just not the right shape. They're not the right structure. As I often point out to students, sometimes you get really cheap vitamins or supplements, and if you look on them, then it won't specify if you have D or L forms of different molecules. So I used to give my previous cat lysine, and I was like, oh, this one's so much cheaper than the other one. And I realized that the other one that was more expensive said that it was the correct form of lysine, whereas the cheap one probably had both of them because it was just made through chemical measures. They just took whatever random thing was made and then sold it. If you're a chemist and you're trying to make only one of these, you either need to make it enzymatically, which is difficult because you need to use organisms, or you just make it through a chemical process and then you have to separate it out, and so then you lose a lot of the material. So that can definitely, makes it difficult and time-consuming and expensive. For us, I don't think that they would. For some bacteria, yes. Yeah. Okay. And so we talk about this again. I also wanted to mention that 10 years ago, maybe more, they found some meteorite or asteroid where they found traces of quote unquote organic molecules, and some of those were amino acids. But again, they usually find both L and D amino acids. So some people say, oh, this is evidence for life, but then biologists will say, well, wait a minute, we would expect there to be more one form than another if these amino acids were made by organisms. So always something to keep in mind. Okay, so why don't we take a short break before we move on to the different types of amino acids? So we'll say like 5 minutes, 5-minute break.
for our little presentations.
I do have them.
I just forgot to write mine exact down, so.
I think I have them in a folder at home.
Okay, so,
But yes, I was going to post that.
okay.
Yeah.
And am I able to change mine?
Probably.
Okay. Because I was just thinking, there's maybe a little more common or relevant, not crazy.
Actually, let me see if I have it in my notebook.
I can always shoot you an email or something too.
Yeah, I just want to, like, if I come here, then I can show it to you. But I can't remember if this is what I did or not. No, I don't have it. Sorry. So I did want to mention that, again, I tried to give this information context, know that the textbook didn't have anything in it about like the nana saccharides, and I don't think the textbook talked too much about the different forms of amylase or things like that. You should know this stuff, at least to the extent which I talked about it, even though it's not in the textbook. So I told many of you who asked that for the exams, always go by the slides rather than the textbook. The textbook is a great supplement, but you should make sure to know what we covered in class. Same thing for the end of this chapter. I'm not going to talk too much about some of the analyses that they talk about, because I don't think they're particularly relevant. So don't go crazy trying to figure out what all these different analyses are in the lab. But then again, on the flip side, we're going to talk about a couple of amino acids that I don't think they talk about in the book very much. So you can go by the slides for that and the recording today. Okay, so we talked about amino acids a couple of moments ago. Again, we said that there is a central carbon. All amino acids have a hydrogen group, an amino group, and a carboxyl group, or sometimes referred to as an acid group. Then finally, the fourth group is called the side chain, or let's see, the side chain, the functional group, or the R group. I think that's all the names I can think of. R group, side chain, or functional group. And what's significant about this? Why do we care about the R groups? Yeah.
Because they identify the amino acids.
Right, so it identifies the amino acid. You have a different R group for every single one of these. And why is that important? Great, function. So they have different chemical properties and different sizes. So the different amino acids will be incorporated into proteins according to their size and their chemical properties. So the one amino acid that we said is not chiral, is glycine. It is the smallest amino acid, and it is not chiral. Does anybody remember why it's not chiral? It has, so a chiral carbon is found for different things. So glycine, what is the R group?
Many commandments. It's just a single atom. I can't remember which one it is.
Can't remember which atom it is. Anybody want to give a guess? The R group is a single atom for glycine. Hydrogen. So super simple. The simplest amino acid that you could have has the central carbon, the hydrogen, the amino group, the carboxyl group or the acid group, and then the R group is just the hydrogen. That's glycine. Again, this is not a chiral carbon because it has two hydrogens, which means that it can be turned around and flipped around. Now, I'm not going to talk too much. Go ahead.
So the side chain is considered the R group.
Yes, so these are all synonymous names. Yes. R group, the side chain, or the functional group. R group, all these can also be used for other molecules, but usually in chemistry. In biology, whenever you use these terms, it's almost always referring to an amino acid. Now, I'm not going to talk too much about the different categories of amino acids. If we talk about anything specifically, then we'll look at the structure and then talk about how it affects the function. Similar to some of those elements which we need in high quantities versus low quantities versus perhaps not at all, for the amino acids, we have some that we as humans can make through various metabolic pathways. And then we have other ones that we cannot make through any of our pathways. So the ones that are starred here are essential amino acids, so they cannot be synthesized. We don't have the enzymes or the pathways to synthesize them. But which amino acids are essential or need to be consumed is different for different organisms. So for the bacteria that we talked about, some of them might be able to make some of these amino acids, whereas they might not be able to make other amino acids. And then we talked about these are the 20 amino acids that we see in most organisms and traditionally, but then there are 3 additional amino acids, at least 3. These are the most common ones. And of course, because bacteria are special, they're the ones who can produce and use these amino acids. So they are FMET, pyrrolysine, and selenocysteine. And these are used for different things. They are incorporated into proteins, unlike the other amino acids, the L amino acids that we talked about, which are not incorporated into proteins. But again, they're only used by certain bacteria. And so we as humans don't have any proteins that require these in their structure. If we consumed these amino acids, it probably would be meaningless. It wouldn't be used in our proteins. So again, the importance of amino acid is that they form polymers, and the polymers that they form are proteins. So when you bring together 2 amino acids, just like with bringing together 2 carbohydrates, you have a condensation reaction and you form a peptide bond. So the peptide bond is the bond between 2 amino acids. And a peptide bond is between a carbon and a nitrogen. So the carbon will have oxygen, and then this nitrogen, and so the peptide bond is this one. And this is a different depiction of an amino acid than we saw on the previous slide, where we were looking at amino acids like this. But remember the components, and so if we take a look at this is alanine, this is the central carbon, then this is the carboxyl group or the acid group. Then this is the central carbon on this other alanine. So it's the same, it's just shown in green. So when we see this peptide bond, does it involve the central carbon? No, right? So I think that's always important to remember. The central carbon is not involved in any types of reactions normally. We're always reacting with the carbon in the carboxyl group in the acid. And then that will react with the nitrogen or form a bond with the nitrogen in the amine group. So again, we're losing the OH from one amino acid and then a hydrogen from the other to produce H2O, making it a condensation reaction and forming that bond between the carbon and the nitrogen. Now this just shows a dipeptide or a peptide with 2 amino acids. We know that most proteins have more than two amino acids. And we could have up to hundreds of amino acids in a single protein. And proteins could be quite large. And proteins come in a huge diversity of structures, sizes, shapes. And so we talk about the different levels of protein structure. They're the primary structure, secondary, tertiary, quaternary. So if we are talking about a mutation in DNA, which then changes an amino acid, what level of structure are we talking about? So if we say the protein had a change in its amino acid structure, some mutation caused by DNA, what structure, what level are we talking about to start out? The primary structure, but which other levels will that affect? Any? None? It could affect all of them. It might not affect any of them. It depends on where it is in the protein, the identity of the amino acid, what was subbed in, if it's a similar amino acid or not. It could make the protein non-functional. It might make it less functional. It could make it more functional, having a higher rate of reaction. It all is specific to the circumstances that you're talking about. So always remember that. So we tend to think of mutations as, destroying the protein. Sometimes thinking that the protein is not even there, but the protein is normally still there even if we have a mutation, but the protein might not work correctly. But the difference there is that if there's a mutation and the protein's still being made, you're not getting the function, but you're still using the resources to make the protein. So it's kind of double bad. You're using the resources, you're using the amino acids that you can use for another protein, but you don't get the function that you need. Okay, so we talked about the primary structure, which is just the order of sequence of amino acids. Then we have the secondary structure. So the secondary structure consists of these kind of familiar organizations of amino acids, the beta-pleated sheet and the alpha helix. So these are both formations. They don't specify any particular amino acid sequence. They're just shapes or configurations of amino acids that are often seen in proteins. The beta-pleated sheet is fairly flat and linear. We often see beta-pleated sheets stacked on top of one another in a protein. And then we have the alpha helix, which can offer stability in a protein. So that is the secondary structure. Then we have the tertiary structure, which is the folded structure of the protein. So that's when you have a 3D protein, you have a lot of the protein formed. And then with some proteins, but not all, you have the quaternary structure, which is where you have multiple subunits coming together. So here's a question. If you have a protein that has quaternary structure, do all the subunits need to be identical to one another? Discuss it with somebody near you for a moment. Or if you don't trust them, don't. Do they all have to be the same? Do we always have to have 4 subunits, like this down here? Any thoughts? No thoughts? Any guesses? Anybody remember hemoglobin? How many subunits does that have? 4. So it has 4, but you don't have to have 4. Anybody remember if all the subunits are the same? It's kind of a tricky one. So the subunits in hemoglobin look very similar, but they are not identical. So there are 2 different subunits, and you have 2 of each in hemoglobin. So the answer here is that subunits do not need to be identical to be part of the quaternary structure of a protein. You could have different subunits. You don't have to have 4. There are definitely lots of proteins that have 2 subunits, but you have many proteins that can have half a dozen subunits. So it really depends on the protein. And interestingly, some proteins will still work, not super well, but they will still work even if you only have the tertiary form. So, for example, if you had something like this might still function perhaps more slowly or at a slower rate than if you had this form.
Subunits are all the same in the first math class.
Can you rephrase? Not sure.
You said the quaternary, the different subunits.
Yeah, okay, so I think I see what you're saying. So this is showing our quaternary structure. We have like this dark, I don't know, pinkish, and then we have this peach color, and then we have 2 yellow colors. So let's pretend that these are all different subunits. So each one of them has a slightly different tertiary structure, a slightly different secondary, and a slightly different primary structure. Or you could have them be identical. It depends on the protein.
On the tertiary and quaternary structure.
So they would all have their own tertiary and secondary structure, but it's not until the quaternary structure that they would have the same structure, that they're part of the same molecule. And you could think about this, you could think about the sequence of how a protein is made. So you get the mRNA, and then you have the mature mRNA, and then you have it being read by the ribosome, you add on amino acids, and then it's folded. And so then you have your tertiary structure. Then if you have other subunits, then you have to have that mRNA, you have to process that. The ribosome reads that, amino acids, then folding, then put the 2 folded ones together, and perhaps that's your quaternary structure.
And the tertiary structure has different secondary subunits?
No. Yeah. Or, okay. A tertiary structure can contain multiple alpha helices and multiple beta-pleated sheets. But for that one specific primary structure, this primary structure is always going to turn into the same tertiary structure. Sorry, I think I was answering a question you weren't asking. Did that clarify? Or we can come back to this too. And if need be, I think I have some videos as well that kind of show the whole process, which I think helps in some cases. Oh, and I also wanted to point out, to complicate things even more, that some of our human proteins that have quaternary structure are really similar so that each subunit is really similar to a certain bacterial protein. So many of our proteins are more complex because they have multiple subunits, but the individual subunits might be similar to bacterial proteins. And we'll see this when we talk about the electron transport chain next week. Okay. And again, I wanted to remind us that form and function are closely linked, so that when we change the primary structure, we often are going to change the other structures as well, and what that protein can do, as we discussed. And so I wanted to just pull from chapter 8 for a moment because I feel like it's related. So chapter 8 is talking about metabolism, and it goes into inhibitors. And I think that putting this right next to that rule that structure and function are related really makes this connection. Because we've been talking about enzymes being really specific to their substrates. And we see here that the active site is similar in shape to the substrate. We know that it would also have a chemistry that is complementary and can interact with the substrate. And then we see that the substrate is worked on, and in this case, there's some bond that is cleaved, and then we get the products being released. Okay. Here, we are talking about cofactor, so we're not talking about inhibition yet, but we're talking about some molecule which can be inorganic. So cofactors can be things like ions, or they can be other proteins or some other inorganic molecule. And that needs to be bound in order for the active site to work correctly. So again, it changes the structure and it changes the function of this enzyme. So oftentimes this term is not used, but the apoenzyme refers to just the enzyme by itself, which is not necessarily active. Then when we have the enzyme plus the cofactor, or the coenzyme, then this whole thing is active, and together both parts are called the holoenzyme. And so only the holoenzyme is active and able to carry out whatever reaction is necessary or is its job to do. So some cofactors that you might have heard of before are things like I'm blanking now. glycolysis and move the pyruvate into the mitochondria. There are 2 or 3 cofactors that act together in order to bind that substrate, and they actually will bring it to the enzyme. So if I recall correctly, there's a cofactor that's like a little spring that will kind of, it bends down to interact with one active site here. This is one. And then the product is here. The product binds to this cofactor, and then it kind of then springs this way, and it goes to the next active site. And this cofactor is really important because it allows the transport of the substrate from one active site to the other really, really quickly. So again, it's in this pathway that is extremely important to metabolism, to making energy. And this cofactor helps that occur much more efficiently. Okay, so then finally, inhibitors. So inhibitors will change the shape of an enzyme. Competitive inhibitors will change the shape by blocking the active site. And then non-competitive inhibitors will bind to another site and change the conformation of the protein. So for non-competitive inhibitors, it's a little bit difficult to imagine for a moment, but I think probably everyone here has seen, oh my gosh, now I forget what these are called. The books, parents here, books that you open and then you pull a little tab and it pops up. Oh, a pop-up. I didn't forget what it's called. So a pop-up book, if you pull that little tab, it changes the structure essentially of whatever the little image is, right? And so that's kind of similar. The non-competitive inhibitors, and pull that tab, completely changes the shape of the enzyme so that the active site is no longer available to the substrate. The competitive inhibitor is a little bit more straightforward. It's just sitting there blocking the active site. So what would you predict about the competitive inhibitor in terms of its structure in relationship to the substrate? In this image, it's smaller. I don't think this is a really good depiction, honestly. Because the inhibitor, the competitive inhibitor, is binding at the active site, which has a certain shape and chemistry. And the substrate also, in theory, can bind at that active site. So we might expect that the substrate and the competitive inhibitor will have similar structures. That is not depicted here. But I think that what they're trying to show is that sometimes competitive inhibitors will be more similar to a transition state. So halfway between the substrate and the product than the substrate itself before it's been going through the reaction. But there's definitely some similarities between competitive inhibitors and substrates because they're binding to the same site.
What would be the percentage of having out a certain site? It would change the enzymes or what kind of what could be productive?
yeah, so there are lots of enzymes. So we're gonna talk about glycolysis next week. A lot of those enzymes are turned off by allosteric sites. Yeah. And so what do we remember about the first few steps of glycolysis? Anybody remember? You gotta spend money to make money. You gotta use ATP to make ATP. Okay. Right. You have to use 2 ATP in the beginning of glycolysis, and you're not getting anything out yet. So if you don't need ATP, you don't want to invest that ATP in the beginning. So a lot of those beginning glycolysis enzymes have some kind of inhibitor site. Yeah. Okay. So we talked about proteins and amino acids. And now we're going to go back to storage molecules for a little while, unless anybody has any more questions about proteins, amino acids, shapes, sizes. No. All right. Okay. So, storage molecules. I mean, we talked about carbohydrates, but usually when we talk about storage molecules, we're talking about fats, right, in our everyday lives. So, Krabby Patties, they have a lot of fat, or so we're told. So fatty acids are these long chains of carbohydrates with lots of hydrogens. We can have saturated or unsaturated fatty acids. So if we have a saturated fatty acid, do we have more or fewer hydrogens? Saturated and unsaturated. So saturated fatty acids, again, we're inferring that there are carbons here. There are hydrogens in every single place where there could be. They're saturated in hydrogens. Unsaturated fatty acids have a double bond, at least somewhere. Some of them have more than one double bond. And so that means that we have a carbon here, 2 carbons, and this carbon has 1 hydrogen, and this guy has 1 hydrogen, and then they're connected to other carbons. So this is not saturated in terms of hydrogens. So unsaturated fatty acids can be referred to as having kinks or bends, whereas the saturated fatty acids are more linear, and they have a tendency to be able to be packed more closely together because, again, they are more linear. Whereas the unsaturated fatty acids, if you have a double bond and the fatty acid is stuck out at a certain angle, then it might prevent other fatty acids from coming together and packing closely next to it. So we've talked about phospholipids in the class before. Phospholipids and fatty acids, they're not interchangeable terms, even though sometimes I think our tendency is to do that. Instead, phospholipids have fatty acids as part of their structure. Other components of a phospholipid are glycerol, and other things that I should mention about fatty acids before we move on is that, are these guys hydrophobic or hydrophilic? Who wants to vote for hydrophobic? Hydrophilic or unsure? Okay, so most of us said hydrophobic, which was a good guess. And these are hydrophobic because, again, these are just carbon and hydrogens, so they don't interact well with water because they're non-polar. So if we look at the structure of complex lipids, like triacylglycerol, so we don't need the dashes, but I put the dashes in there just to help us pronounce it more easily. Triacylglycerols have fatty acids. This is the diagram of it. But then we also have glycerol to it. And then similar to what we were talking about with amino acids, so these are R groups. So it's just some length of a carbon-hydrogen chain, hydrocarbon. So triacylglycerols are molecules for storing energy in the form of fat. And these can be broken down to release energy, but obviously not through the same pathways that we use to break down glucose. So just to make sure that we're clear on that. And in addition, of course, this is somewhat similar to our phospholipids. Our phospholipids only have two fatty acids, two fatty acid chains, but they also have the glycerol. So overall, it's kind of similar to the triacylglycerols. Whoa, what's going on? Okay. So again, I want to bring it back to things that we might be familiar with. So we're always told that there are good fats and bad fats. So different types of fatty acids, so different lengths of the chain, different position of the glycerol and the unsaturated double bonds will interact and stimulate different pathways in your metabolism. And so this just gives some examples of different fatty acids, where they can be found, and how they can be processed. So for example, if we start out with linoleic acid, then that can be converted into gamma linoleic acid, and that can then be converted into this arachidonic acid, which is sensed by your body and can cause inflammation, or at least help to regulate genes and proteins that are related to inflammation. Whereas you're always told that fish oil is a good fat, so fish oil will actually act as an inhibitor for this enzyme that performs this conversion, and in addition, it itself is processed into this other fatty acid. I'm not even going to attempt to read the name. And then that pathway is related to anti-inflammatory products. And so the theme that we're moving towards now is that there are all these different similar organic molecules, and many of them can be converted from one into another. Not all of them. And depending on which ones you start out with, can determine what you do with them in the end. So just like we said with our carbohydrate polymers, if you have certain types of bonds between them, then you need a certain enzyme to break them down. But if you have either starch or if you have help me out here, guys, what we were talking about before. Our glycogen or our starch, then you could use different enzymes and break that down, and then everything in the end gets converted into glucose because we're breaking off glucose molecules. Okay, so other important functions of fats, and again, this is more related to humans than bacteria. But in humans, we have certain vitamins which we need, and many vitamins are actually cofactors, which we just mentioned. And these vitamins are fat soluble, so you're more likely or more able to absorb them if you eat them or consume them with fats. And also, some of these vitamins can be stored in fats. So if you use fat too quickly, or if you're burning fat over time and you have too much of these vitamins stored in your fat, then you can actually release it. And even if you haven't recently consumed large amounts of these vitamins, you're releasing large amounts at once. So this is one concern that doctors sometimes have if you kind of overdose on like vitamin A or vitamin K. So fats are important, even though we try to get a bad rep usually. But they help keep us warm too, so that's important. Because it's not too cold out right now, but it's not warm either, right? Okay, so here we have our phospholipid. And again, we have our glycerol here at the top, we have our 2 fatty acids, and we have our phosphate. So in IntroBio, you probably learned just the stereotypical structure, and you may not have learned that there are different types of phospholipids. We're not going to go through all the different types, but you should know that they exist. The different types are important for different cell types and different membranes. Also, I should introduce a term that we didn't go over last week when we were talking about membranes, and that is amphipathic. So phospholipids are amphipathic. What does that mean? Is that like a real hand raise or a hat?
It has a hydrophilic portion and a hydrophilic portion.
Yes. So amphipathic means that there's both a hydrophilic and a hydrophobic portion in one single molecule. And which part is the hydrophilic part of our phospholipid? The one?
Head.
Yeah, so the hydrophilic head has charges on phosphate group, and so those charges make the head portion hydrophilic, able to interact with water because of its polarity. The tail portion is hydrophobic, again, because it's just carbons and hydrogens. No polarity there, or essentially no polarity. So if we look at a membrane, the outside of the membrane, which is, that is the outside of the cell and the inside of the cell, are associated with the hydrophilic portions. Then inside of the membrane, or in between the 2 leaves of the membrane, we have the hydrophobic portion. So only specific molecules can get through the membrane, and only particular proteins can be inserted into the membrane. And we could also, in addition to the phospholipid bilayer, which we normally see in cells and which we talked about last week, you can also spontaneously form structures like this, micelles, which have a single layer. They tend to be smaller, and no cells have only a single layer of lipids, phospholipids, as a membrane. Now, our textbook also talks about cholesterol, and I'm sure we've all heard about cholesterol as well. So cholesterol is significant to all of your cells because of the cell membranes and the effect that it has on your cell membranes. So this is a really rudimentary chart showing the relationship between membrane fluidity and temperature. So can anyone take a moment and kind of sum this up in a sentence or two?
As temperature increases,
Right. So as it gets hotter, membranes become more fluid. And why is this? What happens in general as the temperature increases?
Reactions increase.
We have increased reactions and increased movement usually. So just the movement of the molecules increases, they're bumping into each other more, and so then you tend to have more fluidity in the membrane. So this can be bad because we want our cell membranes to have kind of a stable fluidity because we like our cells to just stay intact. It's a little bit selfish, but that's how we are. And so the reason for incorporating things like cholesterol into the membrane is because they could help disrupt this relationship. So hopanoids are molecules in the same family as cholesterol, but it's essentially the bacterial equivalent of cholesterol. So they are also molecules that are inserted into the membrane. They are inserted in between phospholipids. They don't span the whole membrane, they just span part of it. And they restrict membrane fluidity into a certain range. And they do that by, at high temperatures, resisting or restricting the movement of phospholipids. So at higher temperatures, these hopanoids or the cholesterol, they're less likely to move around easily because of their structure. They're not linear like some of the tails of the phospholipids. So the phospholipids kind of are held in place by them. And then at low temperature, the hopanoids will prevent the phospholipids from smushing really tightly together. And when they're really close together, that's when the membrane becomes more rigid, and you don't want that because then phospholipids can't move around, proteins in the membrane can't move around, and you're going to have decreased permeability for the things that should get through the membrane. So this is really important for the membrane staying consistent. So this is hopine. A hopanoid has a structure that's kind of similar to cholesterol. If you're familiar, it has ringed structure, and again, as I said, it's not linear. You can see that it has like this bend in it, which is important to its function. And I think that text underplays this, but isoprenoids are the class of molecules that hopanoids and cholesterol both belong to. And isoprenoids are a huge group of molecules that do many, many different things. We can see right here that there's a big difference between the molecules shown here, and they have very different functions. But some of them do function in membranes, some of them function in signaling, and I'm sure we'll run into other functions of isoprenoids in the future.
So just to make sure, so it's a bad thing because things that need to get through the membrane can't anymore.
So, in the case of, why don't we draw it? Because we have a blue and a red marker, so that's perfect. So in the case of cold, if we didn't have our cholesterol or hopenoids, then we would have tight packing. So normally when we're looking at a membrane from the top, we would see proteins, and the proteins could move in any direction amongst the phospholipids. So that's important for the function of the proteins. And then also, what's one really important thing that gets through our membranes? And what else? What else do we need? Even more important than that, just oxygen. So these should diffuse through the membrane, but if they have to go in between the phospholipids, if they're packed really tightly together, then that might not happen, or would happen at a lower rate.
So the bad cholesterol causes our arteries to harden. It's preventing oxygen getting into our bloodstream effectively, or
So that's an
that
additional-
just like caking onto the walls so that blood can't pump as efficiently.
Right, so that's an additional effect, because of the pathways and the accumulation of other things in the arteries. It's not just because of the change in the membrane. So then, in moderate conditions, if there's no hopenoids or cholesterol, there would be excess fluidity. And then we just have, we might have increased permeability and so some things that shouldn't get into the membrane might get through the membrane. And maybe you would have not enough stabilization of the proteins. So relating all this back to bacteria, the text talks about this transporter protein. So it's a transmembrane protein, which makes a channel. It can be open or it can be closed. The configuration changes after this protein is phosphorylated. What does phosphorylation mean? Does anybody remember? It's the addition of a chemical group. What kind of chemical group? It often comes from ATP. Anybody remember? So phosphate groups. So when we phosphorylate, we add a phosphate group. Oftentimes in the images in the textbook, it's just represented as a P, it's a phosphate group. So adding a phosphate group often changes the conformation of a protein. And in this case, it opens the channel. So when we have a phosphate group attached, then we can have chloride moving through this passage out of the cell. And so this particular transmembrane protein is called the CFTR, or the cystic fibrosis transmembrane receptor. And there are different mutations in the gene for this protein. And the book kind of makes it seem like there's only one type of mutation. There are actually several different mutations that you can have, which will either result in a protein in your membrane that doesn't work correctly, or will result in a very low number of proteins in your membrane. And this can accumulate, or this was can start kind of a pathway or a sequence where you end up with the symptoms of cystic fibrosis, and you could also have many times bacterial infections. So the first image here, I'm gonna shut the lights off, or at least on one side so we can see. Yeah. So this first image shows that we have some gene and we're making the CFTR protein. Like I was talking about before, this mutation, the protein is made, but the protein doesn't work very well. And so we see it doesn't look like it's supposed to, and the cell can identify this, so it's kind of degrading a lot of these. It's not even putting them in the membrane. Some of them do get to the membrane, but not all the time. So here we do not actually see any of the transmembrane proteins, the CFTR proteins in this membrane. And this is needed in order to interact with the environment. So in order for these cells to balance the density, essentially, of the mucus on the outside, they need some mucus, but because of the lack of chloride that is being released, the mucus is going to get really thick and build up. So just like in our mucous membranes, obviously we need mucus, but we don't want really thick mucus. Like that is exactly the miserable part of having a cold or something. So in these individuals, these mucus membranes could accumulate a lot of mucus. We often think about it in the lungs, but it's other mucous membranes as well. This affects the cilia, especially in the lungs. So there's airway obstruction because the cilia are not feeding. And in addition, you just have surface covered in this mucus. So this results in inflammation. And then this mucus can also be colonized by bacteria. So we talked about mucus sometimes having certain molecules that are antimicrobial and discourage microbial colonization. But one of the other things that happen here is a change in pH, and I'm not going to go too much into that, but the change in pH of your mucus can be really bad because bacteria will really like that. And so again, all this contributes to bacteria colonizing that mucus. And something I forgot to mention here is that this protein, this transmembrane protein, it has multiple functions. So yes, it lets chloride molecules, chloride ions move through, but in addition, it interacts and controls the function indirectly of other transmembrane proteins. So you're not just changing the concentration of chloride outside and inside of the cell, you're throwing everything out of balance. And that's why these other transmembrane proteins are shown as well. And so that also contributes to the mucus buildup. So one of the most common types of infections of CF patients is Pseudomonas aeruginosa. And this is a bacterium that is just really good at colonizing mucus. And also, we have lots of it all around us, lots of it in our environments, and normally we could fight it off, or our mucus is producing those antimicrobials, so they can't colonize our mucus. The book also talked about this differential media, which shows a color change. So we talked a lot about differential medias last week. So if you grow Pseudomonas on this type of media, you get a bright yellow or greenish color. If you grow other stuff on there, then you're not going to get the color change. So this is a test to see if CF patients have Pseudomonas infection. So when I talk about colonization of mucous membranes by bacteria, what we're talking about is not just a gathering of bacteria, but a change in the lifestyle of bacteria, which makes this even more significant. So for certain bacteria, they have a solitary lifestyle where they use certain genes. But then if they find some good place to colonize, like some more basic pH mucus, then they're going to adhere to that substrate, and then they actually will turn on different genes. So they colonize that mucus, then they turn on different genes, they express different proteins, they might start dividing, and they start sending out signals for other bacteria that kind of say, hey, you should come join us. This is a really good place to live. There are lots of nutrients. So they might share DNA. They might also share proteins. So if they're sending out proteins to break down nutrients outside of the cell, then multiple cells can benefit from that because they're all living together. And then in addition, because this gel is a matrix, then it can also be really difficult for things like antibiotics to get through. So your antibiotics might not penetrate the mucus, and in addition, if any bacteria in this mucus community, this biofilm community, have an antibiotic resistance gene, they can share it easily, because everything's really localized. So you can really start to stack up and get a large population of bacteria in these biofilms. And then eventually they kind of act like a multicellular organism almost, and they send out other bacteria with a solitary lifestyle, which can then go and colonize the biofilm. And so this is the concern for CF patients, because once you get these biofilms forming, it's really difficult to get rid of them. So I think that I had, yeah. So this other example from the book also talked about the Pseudomonas aeruginosa causing a skin infection. So I thought we would watch a short video on Pseudomonas.
Learning medicine is hard work. Osmosis makes it easy. P. aeruginosa is a gram-negative bacteria that is abundant in the environment. It tends to opportunistically infect high-risk individuals. It is well known for its multi-drug resistance, making it hard to treat. P. aeruginosa is an encapsulated gram-negative rod bacterium that's an obligate aerobe, so it uses oxygen for metabolism through cellular respiration. It does not ferment lactose and does not make spores, but it is catalase, citrate, and oxidase positive. It has a flagellum, kind of like a tail at one end for motility, and it has multiple hair-like appendages called pili all over to help with adhesion to other cells. It also has a number of multi-drug efflux pumps that efficiently pump medications out of the bacteria, making it resistant to a variety of antibiotics. In addition, it's able to make beta-lactamases that degrade beta-lactam antibiotics, as well as aminoglycoside-modifying enzymes that alter aminoglycoside antibiotics, rendering them ineffective. In short, its defenses are strong.
And so I just want to mention that not every single strain of Pseudomonas will have all of these things, but they can use these genes to make these proteins.
P. aeruginosa is everywhere in the environment, in soil, in the home, and in hospitals, where it's found on improperly cleaned medical equipment and devices, various surfaces, and on the hands of healthcare workers. It can survive for months on dry surfaces and inanimate objects, but particularly loves humid or wet conditions like hot tubs, contact lenses, catheters, and medical ventilators. Transmission can occur when broken skin or mucous membranes come in contact with contaminated surfaces, or when aerosols containing P. aeruginosa after an infected person coughs or sneezes. Now, a P. aeruginosa infection is actually not too common in the general population. It's an opportunistic bacterium, meaning that it frequently causes infections in high-risk individuals, like those with cystic fibrosis, chronic granulomatous disease, and type 2 diabetes mellitus. It's also common among individuals with immunodeficiency, IV drug abusers, or those with severe burns or deep wounds. P. aeruginosa produces several virulence factors to help it invade epithelial cells and survive an attack from the immune system. First, there's endotoxin, a molecule that's made up of a lipid and a polysaccharide. The endotoxin sits on the outer membrane of the bacteria, and it's recognized by the immune system as being foreign and causes a really strong immune reaction that can cause extensive damage to the host cells. Certain strains of P. aeruginosa also produce a mucoid exopolysaccharide capsule, and this contributes to the formation of a biofilm, a slimy, sticky, gummy material that surrounds colonies of P. aeruginosa and acts like a shield to protect the bacteria from immune cells as well as antibiotics. P. aeruginosa can also produce toxins that can be injected directly into the host epithelial cells through a type 3 secretion system. It's kind of like a fine needle that pierces the host cell and forms a tunnel between the P. aeruginosa and the host epithelial cell, allowing toxins to pass through. P. aeruginosa also releases toxins into the extracellular environment. For example, it releases phospholipase C, which is an enzyme that degrades the host cell membrane, causing the cell to lyse. Other secreted toxins have to move within the cell to act. For example, exotoxin A is released by P. aeruginosa into the extracellular environment and then taken up by nearby host cells, where it targets ADP ribosylation of elongation factor 2, which is a ribosomal protein. That inactivates protein synthesis and eventually leads to cell death. Piocyanin is another example. It's a blue pigment with a sweet, fruity smell like grapes that's released by P. aeruginosa and taken up by nearby host cells, where it generates reactive oxygen species that cause oxidative damage and cell death. In the environment, piocyanin is released by P. aeruginosa, and it's taken up by microbes that are competing for nutrients in space and causes them to die as well. A sweet, fruity death. P. aeruginosa also secretes pyoverdine, a green compound that is an iron chelator that binds to iron and transports it to the P. aeruginosa. Symptoms of P. aeruginosa infection depend on the site of infection. Usually there's a fever, an elevated respiratory and heart rate, and an increased white blood cell count. If the infection is in the blood, it can lead to septic shock. But there can also be localized infections, like an infection of hair follicles called hot tub folliculitis that can look like a red, pimply rash. Burns or deep wound infections have a blue-green color and a fruity grape-like smell, and immunocompromised individuals can develop a skin condition called ecthyma gangrenosum that begins with blisters but rapidly progresses to tissue necrosis. People who use catheters can develop urinary tract infections, and those infections involving contact lenses can develop conjunctivitis and eye pain. Individuals with diabetes can develop osteomyelitis, an infection of the bones, classically from a puncture wound through a moist sneaker insole. It can also cause malignant external otitis, an infection in the ear canal, sometimes called swimmer's ear. Pseudomonas can cause tricuspid valve endocarditis, particularly in IV drug abusers. And finally, individuals with cystic fibrosis can have pneumonia, particularly involving the formation of biofilm. Diagnosis usually involves a complete blood count, as well as cultures of the site of infection, like the blood, sputum, or skin. In terms of treatment, some cases, like Pseudomonas folliculitis, are typically self-limiting, so no treatment is usually necessary. But in more serious infections, cultures and antibiotic susceptibility testing often guides treatment. Topical antibiotics can be used for superficial infections, and aerosolized antibiotics are frequently used for lung infections. In severe cases, especially in immunodeficient individuals, a combination of anti-pseudomonal drugs is needed to overcome multi-drug resistance, like beta-lactam with aminoglycoside, or carbapenems with quinolones and aminoglycosides. So, to recap, P. aeruginosa is a gram-negative rod bacterium that is ubiquitous in the environment. Virulence factors contribute to key features of P. aeruginosa infection, including blue-green color, fruity grape-like smell, and formation of biofilms, which add extra protection to P. aeruginosa on top of its already notable multi-drug resistance. It can cause a variety of diseases from hot tub folliculitis to ecthyma gangrenosum, osteomyelitis, and malignant external otitis, and also causes pneumonia in individuals with cystic fibrosis. Definitive diagnosis is made by cultures, and susceptibility data help identify effective antibiotics. Thanks for watching. If you're interested,
Okay, it is, but it could do all these things, but you have to keep in mind that also we encounter it all the time and we're fine. So it's kind of a balance. Don't get too paranoid. So I'll see everyone for lab on Thursday. Again, please review this chapter so that you're ready to go forward to chapter 8. And I highly recommend reading chapter 8 before we start talking about it next week.
What lab are we printing?
What lab are we printing? I would need to check.
Oh, we figured out what the problem was in the computer lab. It doesn't like edge.